So when I was growing up, homosexuals, injectable drug users, and hemophiliacs had one thing in common. They were dying from a strange, mysterious disease. We finally figured out what it was, but 30 years later with research, we still don't have a cure. I'm talking about HIV. This is important because 1 out of every 300 Americans has HIV today. I'd like to talk about new advances in HIV vaccines and treatment, and to understand this, we need to know what HIV is, how it infects us, and how we can overcome it. So first, I'd like to talk about what HIV is. HIV is a pandemic infection. According to Mann's research in January of 2015, it affects 35.3 million people worldwide, and there are 2.3 million new infections each year. And according to the CDC current of July 2015, there are 1.2 million Americans living with it, and of these, 50,000 will die each year. There are 3 things that make up HIV, and we'll talk about these more in depth in a little bit when we're talking about the vaccines. We have RNA for the genetic core, surrounded by a protein shell, and then there's an outer coating, a slippery fatty envelope. HIV is transmitted through the body, and it can only infect the T helper cell. This is deep in the body in the lymph nodes. This information is according to the National Institutes of Health, September 2015. Now, according to the CDC of June of 2015, HIV can only transfer through body fluids, such as blood or copulatory fluids. Now, when these fluids are outside of the body, say when a drug user shares a needle, it can last for a few minutes, but after an amount of time, 10 minutes or so, HIV will actually break down, and HIV will not transmit through hostile fluids such as saliva or tears because of the acidic content. And contrary to how I was taught when I was a kid, HIV is not transmitted by hugging, holding hands, or being in the room with somebody. Now that we've discussed what HIV is, let's talk about how it overcomes our immune system. So remember that fatty envelope that I talked about? Well, think about HIV as a dog running around in the room biting people, but it's coated in Crisco when we need to grab it. This is very difficult to do. This is an antibody, and each antibody has a grabber, which will grab one specific foreign protein. More insidious is the RNA. RNA is less stable than DNA for genetic information, and to make things worse, there's only one strand, so it is really easy to change the genetic makeup. What this means, according to Gong's research of August of 2015, is HIV can mutate 35% of its genetic information, so not only can it change its shape so our antibodies can't grab onto it, but it can also mutate to infect us more readily. And then we have these blue pieces. These are called the GP41, that means general protein number 41, and they're what grab onto the T helper cell. Upon grabbing on, they'll enter the cell, destroy our DNA, take it over, make copies, and then proliferate. Now that we know how HIV overcomes our immune system, let's talk about the vaccines. So the most promising vaccine is, according to Gong's research of August of 2015, what he wants to do is inject the body with a substance which will grab onto the GP41 protein. This is equivalent to that dog running around the room, getting its head caught in a box, and now it can't bite anybody. Now that it can't bite anybody, we can scoop it out of the body and we can eliminate it. The second way is according to Mann's research of January of 2015. This long thing, the ADCC, is the antibody-dependent cell-mediated cytotoxicity complex. What this means is the whole process of our immune system. Our immune system, the T helper cell, can be thought of as the concierge. If he sees something in the body that he doesn't recognize, he'll call in the bouncer, the B cell, to come out with the antibodies to take out the infection. If we can stimulate the antibody-dependent cell-mediated cytotoxicity pathway to already have antibodies in the bloodstream before we're infected, then when HIV is introduced, it'll be taken out before it can take hold. And a third way that we can vaccinate is by stimulating natural killer cells. Sanchez's research of August of 2015, he used a human analog, he used a macaque monkey. By using these monkeys, we would inoculate females with a topical cream in the copulatory canal, and then after about 1 to 2 hours later, an infected male would come by, and the females would still be protected, almost at 100%. After a 42-month study, these monkeys were protected at about 31% efficacy. That's good, but not good enough. So these are the 3 potential vaccines that are showing the most promise today. In conclusion, we've talked about what HIV is, how it overcomes our immune system, and ways that we can overcome it. A deadly virus does not need to be frightening, carry stigma, nor be a death sentence. If we can work together as a scientific community, we can go forward towards a cure. I'd like to thank you for your time, and I'd like to open the floor for any questions.